going to necessarily grade on how complete it is. It's mostly just a 5-point thing. If you turn it in and you obviously put some effort into it, that's great. But I do need to, well, let's put it this way. The more you give me, the more feedback you'll get, the more you know where you stand grade-wise. So the more you can give me, the better off you'll be. Alright, so we deaminated our amino acids, and we have ammonia now. And so today is the question of where does the ammonia go? And the answer to that question depends on what kind of critter you are. So if you're aquatic, it's most likely that you're going to excrete it as ammonia. If you are a bird, reptile, then you probably excrete it as uric acid. And if you are terrestrial, vertebrate mammals, then you excrete it as urea. Those are your choices. All of them contain less than nitrogen, right? So ammonia, urea, that's the structure you do need to know, is urea. So it's just a carbonyl and 2 amino groups attached to it. And then uric acid if you're a bird or a reptile. Okay, so we need to figure out how to convert our ammonia into urea, so we can get rid of it, because ammonia is toxic to us if it builds up too much. And we're going to do that by the urea cycle. So urea is synthesized in the liver. And then urea is sent out in the bloodstream, where it is collected basically by your kidneys, for excretion in the urine. The overall reaction is 1 ammonia plus 1 bicarbonate plus aspartic acid. And it's going to require 3 ATP to do this. And we're going to get out 2 ADP plus 2 PI plus 1 AMP plus pyrophosphate. And you're going to make urea. Plus this thing. Which hopefully looks a little familiar. Does it look familiar?
Mm-hmm.
No? Yes?
Yeah.
Take your pick. It's a citric acid cycle intermediate. Take your pick.
Right.
Coumarate. In urea, one nitrogen comes from ammonia. And one nitrogen comes from your aspartic acid. And the carbon comes from your bicarbonate. Okay, so really quick look then at the cycle. So this happens on all the steps. You're probably feeling a little overwhelmed with how many pathways and how many steps that you've had to look at about now. And so for this one, know the overall reaction, kind of know some of the intermediates involved, but I'm not going to ask you to draw all these, unless it's something you should already know how to draw, like arginine, right? So this pathway is actually going to start in the mitochondria, and then it ends up in the cytosol, and the cycle is here, and it travels between the cytosol and the mitochondrial matrix. So, step one. Your cycle has 5 steps. Step 1. The enzyme is carbonyl phosphate synthetase. And what you're going to do is you're going to perform a condensation reaction and an activation of the ammonia and the bicarbonate to make the product carbonyl phosphate. And this is going to consume 2 ATP. So up here they're showing that we had our transamination and our oxidative deamination of an amino acid, and then we get ammonia. And then ammonia in the mitochondrial matrix, we're going to react it with a bicarbonate to make carbonyl phosphate, which is this thing right here. That's probably a new functional group that we didn't learn in organic, about carbonyls. Yeah, okay. So it looks like an amide. Technically it is an amide. It's just we're like a primary amide, but you can call that whole group the carbonyl and the nitrogen carbonyl group. Okay, so we're going to look at the mechanism. Don't activate basically the carbonyl so that we can link it together with the ammonia. And so to do that, we use ATP to activate it. And so we get a little methyl substitution there. And then this is the activated form of the bicarbonate. And then ammonia can act as a nucleophile and come in and splice it. And you make the carbamate intermediate. And then we have to activate the other side too. So another ATP comes in, and we activate it by adding the phosphate on there, but the mixed nucleophilic substitution doesn't happen. So this is the end of our first step to make the carbonyl phosphate. How would you classify that enzyme? Why are you saying ligase?
It consumes ATP.
Consumed ATP and
Made a bond.
Made a bond.
made a bond between the two things. Okay, so ligase. Okay, step number 2. Ornithine trans carbamyl ase is the enzyme. This transfers a carbamyl group which is basically this. To ornithine to make citrulline. These first 2 steps happen in the mitochondrial matrix. The rest happens in the cytosol. Ornithine is an amino acid, but it's not one of the standard 20. Ornithine is right here. What does it look like? It looks like lysine. Yeah, it looks like lysine. It's missing 1 carbon in its side chain. So ornithine comes in. I have my carbamyl phosphate, and I'm going to add the carbamyl group to the end nitrogen in the side chain of ornithine, and that makes citrulline, which is also technically an amino acid, but not one of the standard 20. And hopefully this is easy enough. How would you classify ornithine transcarbamylase? Yep, that's right. So it's kind of animated, right? It transfers a carbamyl group. Transferase. What's the chef that always says BAM? Who is that?
Alazar.
Bam?
Emerald? Yeah, Emerald. Yeah. Because I feel like Emerald when I get to this section.
I love Italian. It's the best.
succinase, sorry. Enzyme. Succinase. Nope, I skipped one. I was right. Anyway. Sorry. Succinate. Synthetase. That was the next one, number 4.
Is it arginine?
It is arginine. This is condensation of citrulline to aspartate to make arginosuccinate. Succinate. It consumes 1 ATP, so 2 ATP equivalents, because it takes ATP all the way down to AMP. Okay, I didn't bother to ask you this one, but what is it? What class?
Ligase.
It's a ligase. How do you know?
ATP.
Join 2 things together. You can usually tell by the names. So when it's a synthetase, those are usually ligases, or supposed to be ligases. When it's a synthase, then it's something that also is making something, and it might be bringing 2 things together to do that, but it doesn't consume ATP just to join 2 things together. So most people are careful about that when naming the enzymes, but occasionally you'll see them swap it because they aren't being careful. Just warning. But synthetase is usually the case. Okay, so here's another mechanism that you do not need to learn, but we'll just look at it quick so you can see that it is indeed ATP being used, AMP being off. So ATP, and I release a pyrophosphate, and I'm adding this AMP group to the carbon in the carbonyl group. When you see something like this, it's always an activation step. I'm activating that group so it will react the way I want it to there. What I created here, what is that?
Amino.
It's an amino. It had positive charge on it. What have we been calling that in mechanisms? What have we been calling Schiff bases? Electron sinks. Yeah. So the whole point of activating this is to create that electron sink and to make this carbon, because the electrons are going to be kind of pulled that direction, and so I can actually get this aspartate to attack that carbon and join them together. So first you activate it to make this more reactive, and then the aspartic acid attacks, and then he leaves, and I'm left with this thing, which is just weird, argininosuccinate. I started with something that kind of looked like lysines that was 1 carbon short. I stuck that carbonyl group on it, and then when I bring in the other nitrogen, I now have what is referred to as a guanidino group, which looks like arginine, right? Arginine has a guanidino group and a side chain. But instead of that just being a free nitrogen, I've got this another amino acid attached. Let me go back to the slide again. So again, here we are, we're in the mitochondria, carbonyl phosphate, then we transfer the carbonyl group to citrulline. Citrulline leaves at that point. So now we're in the cytosol for this last reaction. So step 3 is in the cytosol, and the aspartic acid comes in, and we make arginine succinate. aminosuccinase. This is elimination of fumarate to make arginine. I suppose you could also think of it as the elimination of arginine to make fumarate. Either way. Or vice versa. Fumarate is a CAC intermediate that can also be converted to oxaloacetate. For gluconeogenesis. So it's not wasted. So we're trying to get rid of our ammonia waste product, and we had to consume another amino acid to do that. But the skeleton of an amino acid ends up going right back into the CAC, or gets used for gluconeogenesis. So here's my argininosuccinate. I'm going to lose the fumarate, and they nicely color code this for you. So you can see that fumarate came entirely from the aspartic acid that came in. But it's going to leave behind this nitrogen group because ammonia, remember ammonia only provided one nitrogen in my urea, and I have to pick up another nitrogen from another source. And that comes from aspartic acid. So fumarate leaves, and when fumarate leaves and leaves behind its nitrogen, then I have arginine, and that should be a familiar one. Sorry, this is going to be I hope an easy question. How would you classify argininosuccinase? Two are holding strong. It is a lyase. Yeah, lyase. Well, if I use the word elimination, then that probably is a hint there, but you can think of it as here. You can say that you're eliminating fumarate, but you could also say that you're eliminating arginine, and if you're eliminating arginine, you just form a carbon-carbon hold on in the process as well. So you can think of it that way, that I'm eliminating the whole arginine and hydrogen from the side where you form the carbon-carbon double bond. But I wrote it that we're eliminating fumarate because it's the arginine that continues on in the cycle. Okay, moving on. So, 5. Arginase is the enzyme. And this is hydrolysis of arginine to make urea. And then to complete our cycle, we have to regenerate ornithine. So last step, here's my arginine, and if water were to come in and attack this again would just be another nucleophilic acetyl substitution going on, although rather than an acetyl, it's an amino instead. But water comes in and attacks, and I end up losing the ornithine part of it, and then I have my urea that gets formed. Okay? And then ornithine can come back into the mitochondrial matrix and it's ready to pick up another carbonyl group. Urea cycle. Good?
Good.
So know what the species are involved by name. I won't ask you to draw them other than the ones you ought to know. Urea, fumarate, you already know. Aspartate, you already know. Arginine, you already know. So it's not too bad. Sorry. It's not too bad. Regulation of the urea cycle. This happens at the first step. Of our carbonyl phosphate synthetase. It has an allosteric regulator. That is N-acetyl glutamate. And it is an activator. Now, you know the structure of glutamate, so you could probably draw this one on the end. You know what an acetyl group looks like, so you just need to know where to attach it. So the acetyl group goes to the nitrogen. N-acetyl is linked through the nitrogen, or glutamic acid. Why would glutamate be a good sensor for the urea cycle?
That's a very good question.
Yes, it is. That's a very good question. Let's go back to all the way back
in here
to
somewhere.
all the way back to there. Okay. So it's not in the urea cycle itself, okay. But remember all our alpha keto acids?
Oh,
Alpha
yeah,
keto
yeah.
glutarate always picks up the amino group from the other amino acid you're trying to break down. And so glutamic acid is always produced in that first transamination step. Okay, so if you were breaking down proteins, so you're breaking down amino acids for fuel, what you typically see is an increase in transamination. And that's due to using amino acids as fuel. That causes an increase in the concentration of glutamate. And an increase in glutamate causes an increase in concentration of N-acetyl glutamate. Not a reaction that we're really going to do, but you've got an enzyme that works just based on substrate availability, that converts glutamate into the N-acetylglutamate. And that is an N-acetyl glutamate synthase. Okay. That one is not a synthetase, that is a synthase. So it's not a ligase. What is it going to be?
I'm sorry, N-acetyl, what's that word? I don't know if it is.
N-acetyl glutamate synthase.
Okay.
What happened to it? It's not a ligase, but it now has an acetyl, whatever you want to call it, acetyl, whatchamacallit, group attached to it. Transferase, yeah. Don't tell me where that came from, don't worry about it. Okay. So, more transamination happens, more glutamate around, more acetyl glutamate gets formed, that one goes to the carbamyl phosphate synthetase and upregulates it, because if you've got lots of glutamate being formed, then you've got lots of ammonia that is going to get released when it gets deaminated. Okay? All your other enzymes are dependent on substrate availability. Okay, so we have urea, liver sends it out to the bloodstream. Okay, we figured out what happened to our fumarate, but we got some other things to consider. So this is fate of alpha keto acids. And so we have all these different amino acids that we can break down for fuel, but they don't all go to the same place initially. So there are lots of different fates. Most of these are going to enter metabolic pathways. But at different spots. Depending upon the amino acid structure. Most become citric acid cycle intermediates. Some become citric acid cycle substrates. Those are different. Okay. What's an example of a substrate for the citric acid cycle? Yeah, so acetylcoenzyme A. So that is a substrate, right? And if you go back a step, you can get to pyruvate. They're coming from glucose. But the acetylcoenzyme A is going to come from fatty acids, and they'll converge there, and then that is a substrate that gets oxidized by the citric acid cycle. Whereas things like fumarate is a citric acid cycle intermediate, or it can be converted into something that conventionally is a substrate. Okay. Some can become substrates for gluconeogenesis. So some can be used to synthesize glucose, but not all. some can become substrates for ketone bodies. And there are 7 possible fates. One gets converted to pyruvate. 2 gets converted to alpha ketoglutarate. 3 gets converted to succinyl coenzyme A. 4 gets converted to fumarate. 5 gets converted to oxaloacetate. These first 5 come from what we call the glucogenic amino acids. And 6 is acetoacetate. And 7 is acetylcoenzyme A. And these last 2 come from what we call ketogenic amino acids. Glucogenic, these are amino acids that can be used as precursors for glucose. Ketogenic, these are amino acids that can be used as precursors for ketone bodies or fatty acid synthesis.
Do you mind moving that?
Yep. Okay. So, the simple diagram. I have my amino acid, I deaminate it. The ammonia gets thrown into the recycled and I can excrete it. What I'm left with is the alpha keto acid, and that is going to get shunted into different places, but if I am not just using it as a citric acid cycle intermediate, I can either use it as a substrate where it gets oxidized for fuel, I can use it to synthesize glucose to send back out into the bloodstream, I can turn it into acetylcoenzyme A and become a substrate for the citric acid cycle there, or I can use it to synthesize fatty acids that we can then go store as fats in adipose tissue, or I can convert it into a ketone body if I'm starving and send it to my brain. Okay? So those are the choices. The amino acids feed into lots of different things. And I think it's incredibly useful for you to know what every single amino acid can become. The chapter spends a lot of time on that, going through every single possibility. We're not going to cover any of that. We don't have time. So we are kind of ending here on the chapter 21 material. There are some general things that I would like you to know, and that is mostly what is ketogenic versus what is glucogenic. So lysine and leucine. These are ketogenic only. And then a bunch of these more complicated ones. Isoleucine, threonine, phenylalanine, tryptophan, and tyrosine can be either ketogenic or glucogenic. And they can enter into metabolic pathways in more than one place. So they can be used for either synthesis of glucose or synthesis of ketone bodies or fats. Okay, some general trends that we can pull out of some of these. What do those amino acids have in common? Anything? And it might be more than one trend. What do those three have in common?
Benzene.
They're aromatics, okay. So you can kind of remember, oh yeah, my aromatics, not missing, yeah, but my ring ones. Yeah, benzene rings in them, those can be either. Lysine and leucine, that's a little harder to remember, and when we talk about what's essential and what's not, we're going to see that branch things tend to be harder to make. But lysine and leucine, you remember those ketogenic only. If you remember your aromatics can be in either, then you're kind of left with the isoleucine and threonine is also going to be either. All the rest are glucogenic only. But we're not going to go through all the different pathways and how everything funnels into some metabolic pathway. Just like that. Okay. The other thing that we're going to spend very little time on, all of 5 minutes, is the reverse, kind of like we did. We didn't spend much time at all on the fatty acid biosynthesis. We looked at it, but we didn't go on it. Amino acid biosynthesis. We're not going to do any reactions or mechanisms. We're just going to talk about some generalities. You've probably heard of essential amino acids. And these are amino acids that cannot be synthesized by mammals. I say us. But it must be acquired in the diet. If we can synthesize it, it is non-essential. And that is synthesize it from other sources. Anyone in here taking nutrition? Yeah. How many of you are pre-med? So, no, they'll teach that to you in medical school, so it doesn't matter.
Not even all of us.
That's something I've heard in nursing school. Essential amino acids in humans. Anybody know one?
Phenylalanine, I think.
Phenylalanine?
Phenylalanine. I think it's, I know there's an acronym for it. I think it's,
There's an acronym.
probably,
Is there?
I think. There's 10 total, right?
Kinda. No, we'll talk about that. Any guesses?
An acronym for the
Glutamate.
Cysteine? one wins this? Okay, so
Out here.
Just start.
Purity.
Acidity technically is not, but nutritionists call it that because the synthesis is really slow. There are
Glutamate.
technically 8 essentials. 10 if you don't know better. They include your branched amino acids. So which amino acids are branched?
Isoleucine.
Isoleucine.
Really?
Valine?
Leucine?
This one I don't have a good rule for. Methionine is an essential. It's sulfur containing. But that isn't a good rule to use because cysteine isn't essential. But as long as you have one sulfur containing amino acid in your diet, if you have methionine in your diet, you can make the cysteine. Your aromatic amino acids, but it's not all of them. But getting that benzene ring initially is hard. We can't synthesize that. Take a guess. Aromatic. You got 2 out of 3 possibilities. Phenylalanine. You gotta have phenylalanine in your diet,
Tryptophan.
and you need tryptophan in your diet. Tyrosine can be made from phenylalanine, so tyrosine is an essential.
Well, Sharon just said something like, well, why can't we make methionine from the other sulfur container? Why can't we make
But you don't have enzymes that do that.
Okay.
Okay.
The methionine is the essential cysteine, is not. Okay, so we have the enzymes to make cysteine. Okay, also, there's no rhyme or reason to this. Lysine, you can't make, you have to have it in your diet. And threonine. You can almost think of threonine as a branched. Branched things are hard to make. But it's not really branched, but the hydroxyl groups are in different place. Okay. So those are the 8. Last 2 aren't really essential. The biosynthesis is slow. So if you lack it in your diet, you will feel debilitating effects from it if you don't have these. Even though we can make them. And that is the histidine and arginine. Okay, so if you're a nutritionist, you lump those in with the essentials. If you want to be healthy, you need to have that in your diet. So, like I said, we're really only kind of touching on a couple topics in that last half of chapter 21, past the area central. There's not a whole lot there, so don't spend a ton of time reading through that, although it's good for you. You should read it. It won't be on the okay. We're going to do chapter 22.